So you are able to see my screen? Okay. So once again, good morning to all of you. So we have been studying about transport across membrane. In the previous class, we have studied about the structure and functions of plasma membrane, how plasma membrane is important for the transport of so many molecules across the plasma membrane, whether it is inside the cytoplasm or outside the cytoplasm. The transport can be from the ECF to the ICF or from the ICF to the ECF. So in the previous class, we have studied about passive transport. So what is passive transport? It is a kind of transport where the movement of the molecules does not require any energy. That is a very important thing. And the main difference between the passive and the active transport is the utilization of energy, whether is energy is utilized or not. In passive transport, the transport of molecules does not require any energy. It will just be transported across a gradient. The gradient can be a concentration gradient where it depends upon the number of molecules on one side of the membrane and the other side of the membrane. The difference in the concentration gradient. Okay, the difference in the concentration across two sides. Okay, whether it is been separated by a membrane or not, when there is a difference in the concentration, it forms a gradient. If the concentration is the same across the two sides, there is no gradient. Only when there is a difference in the concentration, it forms a gradient. The gradient may be a concentration gradient where there is a difference in the number of molecules or concentration of molecules or solute. Whereas then there is a difference in the electrical potential. Okay, what do you mean? The charged molecules. Then it forms an electrical gradient. So the combination of both the concentration and the electrical gradient will give you another term called as the electrochemical gradient. So the molecules will diffuse. Okay, there are two types, diffusion and osmosis. In diffusion, the molecule will diffuse. The molecules, what I mean is the solute molecules diffuse from a region of higher concentration, whether it is a concentration gradient or an electrical gradient or electrochemical gradient, downhill to a region of lower concentration. So that is what is happening in passive transport. So the diffusion can be of three types. What are they? It can be a simple diffusion or it can be a facilitated diffusion. So facilitated diffusion, it can be that is a channel mediated diffusion or carrier mediated diffusion. Another process is called as osmosis. So you should clearly know the difference between diffusion and osmosis. Diffusion is the transport of molecules, whereas osmosis is the transport of water. Okay, you should clearly understand. So in diffusion, whether the two regions are separated by a membrane or not, diffusion occurs. So diffusion, the other name for diffusion is just spreading. Okay, just you have to imagine or take an example of mixing a spoon of sugar in a cup of tea, or mixing a spoon of salt in water or lime juice or hot water. So there are many factors we have seen, the temperature, the surface area, the concentration, and other factors. Okay, which will either increase the rate of diffusion or decrease the rate of diffusion. So whatever it is. Okay, so this diffusion is the spreading of the molecules, spreading of the solute. Okay, from one region to another, from a region of higher concentration to a region of lower concentration, till an equilibrium is reached. Whereas osmosis is the transport or movement of water from a region of higher concentration of water to a region of lower concentration of water, not the diffusion or transport of solute. So this you have to make very clear. Now we will come to active transport. So active transport, now you would have known a clear difference between active and passive transport. Passive transport does not require energy, whereas active transport requires energy. Okay, so some polar or charged solutes that must enter or leave the body cells cannot cross the membrane, plasma membrane, through any form of passive transport because they will have to move uphill. So we have seen the transport of sodium, potassium. In the previous class, we have seen about the transport of potassium. Okay, but this is a different case where the molecules the charged molecules are the polar. So in passive diffusion, we know that the lipid soluble, because plasma membrane is a lipid layer, okay, phospholipid bilayer, only the lipid soluble non-polar compounds can easily pass through. But the polar or charged molecules cannot pass through the plasma membrane by simple diffusion. So for the transport of the polar or charged molecules, the passive transport will not be helpful. So it is a movement called as the uphill movement. Okay, what do you mean by uphill movement? The movement or the transport of the compounds or the polar or charged molecules from a lower concentration to a region of higher concentration. So this is called as uphill movement. So in the last class, I have told you how you push the stone from the bottom of the hill to the top of the hill. So it requires a lot of energy. Okay, it cannot be done just like a downhill process, what you have to just push a stone. Okay, that is enough, it will automatically come down the hill by rolling. But the uphill transport requires energy, so they cannot be transported through passive transport. So that the process by which the polar or charged molecules to be transported in an uphill gradient is called as active transport. So why it is called as active transport? Because the name active itself shows that, or emphasizes that it requires energy. Active, okay, it has to be actively transported. So it requires energy. The protein molecules which are involved in the transport, just like the facilitated and the carrier proteins which are present embedded in the plasma membrane, requires energy to move a solute across the plasma membrane against a concentration gradient, uphill movement. So there are two kinds of cellular energy can be used. Okay, two sources of cellular energy can be used for this active transport. So you know the cells acquire energy by the oxidation of food substances. Okay, oxidation of food substances by glycolysis CCA cycle. Okay, so number one is energy is obtained from hydrolysis of ATP. So ATP is acting as the primary source for energy in the active transport. The next form of energy is stored in an ionic concentration gradient. That is the secondary form of energy. Okay, that is, for example, the sodium potassium pump. Okay, so the energy stored in the sodium potassium pump, which is already doing some process. Okay, that is the concentration gradient, which the energy, some energy will be stored in the ionic concentration gradient. So that will be used as energy instead of ATP, or the previous hydrolysis of ATP would have left some energy. So that energy can be utilized for the means of energy that is stored in the ionic concentration gradient for this transport. So now you have to understand clearly what kind of molecules are transported through active transport. So solutes actively transported across the plasma membrane include several ions. So what are the ions? Sodium, potassium, hydrogen, calcium, iodide, and chloride ions. Okay, so these are the ions. So you would have studied about sympoters and antipoters, and you would have studied many examples. Sodium potassium pump. Okay, that is the sodium potassium ATPase. Okay, hydrogen pump. Okay, hydrogen ATPase. Calcium pump. Okay, calcium ATPase. So it is called as a pump, and also and the name of an enzyme ATPase. So it acts both as a pump as well as an enzyme. Okay, so this enzyme part of the pump will act in the hydrolysis of ATP. So the hydrolysis of ATP only gives the energy. So for the purpose of supplying energy, it acts both as a pump, which will help in the transport of the molecules, and also as an enzyme, so that the ATP molecules can be hydrolyzed to provide energy. So the ions like sodium, potassium, hydrogen, calcium, iodide, and chloride can be transported through active transport by processes called as sympoters, where the two molecules are transported towards the same side, and antipoters, where two molecules are transported on different directions. So not only the ions, some amino acids and monosaccharides also can be transported. So this is very important. Now. The transport of amino acids, glucose, they are done only by active transport. So you would have studied during digestion how the glucose molecules and amino acids are absorbed by the bloodstream and transported. So this involves mainly the active transport. Okay, that is mainly antiporters. So not only in the process of digestion, that is absorption, and supply of these or distribution of these molecules to the individual cells, even in the kidney, this active transport plays a very important role in the reabsorption of glucose and amino acids. Okay, that is also done by active transport. Now, how many types of active transport are there? There are two types of active transport which utilizes the two forms of energy which we have discussed a little earlier. What are they? They are primary active transport and secondary active transport. Now, what is primary active transport? Here, energy is derived mainly from the hydrolysis of ATP. So this energy will change the shape of the protein, carrier protein. So a carrier protein which is present in the plasma membrane, that is, it is called as the intrinsic protein, acts as a pump. Okay, it acts as a pump so that it pumps the so pump nale, you can imagine a water pump. Okay, nowadays you don't pump any water. Before, there was a hand pump which you have to pump water from the ground, so it requires energy. Okay, for pump nale, a human being has to pump, use his hands to pump the what do you call the pump. Okay, the water pump, so that it by a process called vacuum and suction, it sucks the water from the underground and gives the supply of water. Okay, just like that, here also the name pump itself denotes that it requires energy in the form of ATP. So this primary active transport utilizes the energy from ATP, okay, by hydrolyzing the ATP, and it will help in the pumping of the an ion, okay, a molecule or a substance across the plasma membrane against a concentration gradient. So the carrier proteins are called as pump. Okay, not only they are called as pump, they are also called as ATPase. Okay, it has got another name. Okay, it is the enzymic name. So the sodium potassium pump acts as an sodium potassium ATPase because this pump hydrolyzes the ATP. So that is why, because it is acting in the or catalyzing the hydrolysis of ATP, it is called as ATPase enzyme as well as a pump. So what is the role of the sodium potassium pump? Sodium potassium pump is a very good example for primary active transport. So this particular pump expels the sodium ions from the cells and brings potassium ions inside. Okay, so transport of sodium from the cells to the extracellular fluid and transport of potassium from the ECF to the cytosol. So this is the action or role of the sodium potassium pump. So why is the sodium potassium pump very important? So this sodium potassium pump maintains a low concentration of sodium in the cytosol. Okay, so what does it do? It will pump the sodium, expels the sodium ion from the cell. So the concentration of sodium and chloride is more in the ECF than the NCE and the ICF. So the concentration of sodium and the chloride should be maintained low inside the cytoplasm. So that is done by the sodium potassium pump. Okay, it pumps the sodium outside the cytosol into the ECF, so that the concentration of the sodium is maintained low inside the cytosol and more in the extracellular fluid. So a concentration gradient is maintained across the plasma membrane where the sodium concentration is more outside the plasma membrane than the inside. At the same time, what does it do? It will move the potassium into the cells. So this is against the potassium concentration. The previous passive transport, you have seen that there is another pump, which is specially called as the potassium pump. So what is the role of the potassium pump? Is to transport potassium from the cytosol to the outside. So here, what does it do? It is this particular pump simultaneously transports the sodium outside and potassium inside. So the potassium concentration in the cytosol is more than the concentration of potassium in the ECF. So this also has to be maintained. For that, this pump works in a manner where it pumps the potassium into the cells against the concentration gradient. Okay, that is why this requires the energy in the form of ATP. So this sodium potassium pump, there are thousands and thousands of sodium potassium pump in a single cell. ஒரு செல்லுலேயே நிறைய சோடியம் பொட்டாசியம் பம்ப் இருக்கும். Okay, if the number of potassium pumps are low or not functioning, then it will affect the homeostasis of the body. So they have to work non-stop. Okay, only then the concentration of the sodium and the potassium will be maintained. Okay, so low concentration of the sodium and high concentration of potassium in the cytosol. So this will be done only by the help of the sodium potassium pump. I hope you understand. Now you can see the process, okay, of the sodium potassium pump. See, you can see the green color arrow and an orange color arrow. So this arrow shows the gradient, okay, sodium gradient. So sodium concentration is more on the outside, okay, than the inside, okay, whereas the potassium concentration is more on the inside than the outside. So this is the gradient. Now the what is the role of the pump? So the pump works against the gradient, uphill. So it has to transport the sodium from the cytosol into the ECM. In the cytosol, the gradient is very low. The concentration of sodium is very low. But still, it has to transport whatever sodium is available in the cytosol from a region of low concentration to a region of high concentration ECF. Okay, that is the role of the pump. Similarly, the concentration of potassium is see from the it is more here. Potassium is concentration is more in the cytosol in the ICF when you compare it with the ECF. But still, it has to bring the potassium from a region of lower concentration, that is the ECF, into the cytosol. So this is against the concentration gradient. So this pump, how does it work? This is called as the pump, and this is the otherwise called as the sodium potassium ATPase. Okay, so what does it do? See this when ATP binds to the sodium potassium pump. Okay, see first sodium enters from the cytosol, and it is present in this region. Okay, so when ATP binds to the pump, it is split into ADP and PI. So now the PI phosphate binds to the pump. So this will enable the opening of the channel to the outside. So the three molecules of Na plus are expelled out. So after the expulsion of the three molecules of sodium, two molecules of potassium enters and binds this. Okay. So when the potassium leaves the pump, the phosphate leaves the pump, the potassium can be released or imported into the cytosol. This is how it happens. So the potassium pump in one transport expels three molecules of sodium from the cytosol to the external fluid, extracellular fluid, and it imports three molecules of potassium into the cytosol. So this is how it happens. So what are the different stages? The operation of the sodium potassium pump. How does it work? Okay, three sodium molecules are transported into the cytosol. Okay, so what happens? The first step, as you have seen in the previous picture, three molecules of sodium bind to the potassium. To the pump. Okay, where does it bind? It binds in the region of the cytosol. Now the binding of the sodium will trigger the hydrolysis of ATP. Okay, after it binds, ATP will come and bind to the pump, and this will trigger the hydrolysis of ATP into ADP and phosphate. Okay, so this phosphate that is being detached from the hydrolyzed from the ATP will bind to the pump. Okay, will bind to the protein pump. Now this binding of the phosphate to the pump will create a chemical reaction and will change the shape of the pump. Okay, so when the shape of the pump is changed, the sodium which is trapped in the pump can be expelled into the extracellular fluid. Okay, now because of this shape, what will happen? It favors the binding of the potassium from the extracellular fluid to the pump. Now this binding of the potassium, what will happen? It will trigger the release of phosphate. Okay, so binding of sodium will trigger the attachment of the phosphate, whereas the binding of the potassium will trigger the release of the phosphate from the pump. Now again, because of the release of the phosphate from the pump, it causes a change in the shape of the pump, which will allow the pump to come back to the original shape and release the potassium that is being attached to the pump into the cytosol. Okay, so again now the pump starts working again. The same cycle repeats. Okay, same cycle repeats again and again. another 3 molecules of sodium will be ready to get attached or join with the pump. Okay, and then ATP will be hydrolyzed, P phosphate will be attached to the pump, so that there is a change in the molecular shape of the pump, which will release the sodium outside. Okay, once the sodium is released, 2 molecules of potassium will bind to the pump. When the binding of the potassium happens, the phosphate is released, which will again create a change in the shape of the pump and releases or imports the potassium into the cytosol. Did you understand? So this is the role of the sodium potassium pump. So you have to clearly understand that the sodium potassium pump works against the concentration gradient. So sodium concentration எங்க அதிகமா இருக்கும்? ECF ல. potassium concentration எங்க அதிகமா இருக்கும்? cytosol ICF ல. ஆனா இந்த பம்ப் எப்படி வேலை செய்யுது? சோடியம் வெளியில தள்ளுது. எங்க இருந்து வெளியில தள்ளுது? சைட்டோசால்ல இருந்து எக்ஸ்ட்ரா செல்லுலர் ஃப்ளூயிடுக்கு வெளியில தள்ளுது. ஓகே, ஆக்சுவலா சைட்டோசால்ல எப்படி இருக்கு? கான்சன்ட்ரேஷன் கம்மியா இருக்கு. ஸோ கம்மியான கான்சன்ட்ரேஷன் இருக்கிற இடத்துல இருந்து அதிகமான கான்சன்ட்ரேஷன் இருக்கிற இடத்துக்கு இது வந்து டிரான்ஸ்போர்ட் பண்ணுது. Okay, so you have to clearly understand this is the concentration gradient, that is the uphill movement or uphill transport from a region of lower concentration to a region of higher concentration. Okay, that is how the level of the sodium will be maintained high in the ECF and the level of the sodium will be maintained low in the ICF. புரியுதா? நம்ம சைட்டோசால்ல சோடியமோட கான்சன்ட்ரேஷன் லோவாவே மெயின்டைன் பண்ணணும். அதுக்கு தான் இந்த பம்ப் ஹெல்ப் ஆகுது. அதே மாதிரி பொட்டாசியமோட கான்சன்ட்ரேஷன் எங்க ஜாஸ்தியா இருக்கு? செல்லுக்குள்ள ஜாஸ்தியா இருக்கு. ஆனா இங்க டிரான்ஸ்போர்ட் எப்படி நடக்குது? வெளியில இருந்து உள்ள அனுப்புது, உள்ள டிரான்ஸ்போர்ட் பண்ணுது. So that the concentration of potassium is low in the maintained low in the ECF and maintained high in the ICF. So this you have to understand clearly. புரிஞ்சுதா எல்லாருக்கும்? Yes or no?
Yes, ma'am.
Okay. So next, what is the importance of the sodium potassium? இதனால என்ன இது? செல்லோட வால்யூம், செல்லோட கான்சென்ட்ரேஷன் வந்து மெயின்டைன் பண்ணுது. ஃபார் மெயின்டைனிங் நார்மல் செல் வால்யூம். ஏன்னா இந்த கான்சென்ட்ரேஷன் ஆஃப் அயான்ஸ் மெயின்டைன் பண்ணாதான் வாட்டரோட வால்யூமும் மெயின்டைன் பண்ண முடியும். வால்யூம்னாலே யூ ஹாவ் டு அண்டர்ஸ்டாண்ட் தட் இட் இஸ் டீல்ஸ் வித் வாட்டர். ஓகே, ஸோ வாட்டரோட கான்சென்ட்ரேஷன் மெயின்டைன் பண்ணணும்னா அயானோட கான்சென்ட்ரேஷன் ப்ராப்பராக மெயின்டைன் பண்ணணும். நம்ம சொன்னோம் இல்லையா, டோனிசிட்டி, லாஸ்ட் கிளாஸ்ல டோனிசிட்டி பத்தி பார்த்தோம். ஓகே, ஸோ கான்சென்ட்ரேஷன் ஆஃப் த சொல்யூட்னா இந்த அயான்ஸ் எல்லாம் சரியா இல்லைன்னா வாட்டர் வந்து அதிகமான கான்சென்ட்ரேஷன் ஆஃப் வாட்டர் இருக்கிற இடத்துல இருந்து லோவர் கான்சென்ட்ரேஷன் ஆஃப் வாட்டர் இருக்கிற இடத்துக்கு போயிடும். ஓகே, ஸோ எங்க it would also attract, that is when the concentration of solute is more, it will attract more water. Okay, it will attract more water. When the concentration of solute is more, the volume of water will be less on that side. புரியுதா? So on the opposite side la volume of water will be more. So by a process of osmosis, the water will start flowing from a region of high concentration of water to a region of low concentration of water. Okay, எப்படி அது low concentration of water இருக்கும்? So solute or the ions ஜாஸ்தியா இருக்கும்போது வாட்டரோட லெவல் கம்மியா இருக்கும். ஓகே, ஸோ இந்த பொட்டாசியம் பம்ப் இல்லைன்னா இந்த செல்லோட நார்மல் வால்யூம், நார்மல் கான்சன்ட்ரேஷன் மெயின்டைன் பண்ண முடியாது. நம்பர் 2, இந்த சோடியம் பொட்டாசியம் பம்ப், இது சோடியம் பொட்டாசியம் எல்லாம் சார்ஜ்டு மாலிக்யூல்ஸ் இல்லையா, Na+, K+. ஸோ இந்த பம்ப் வழியாக தான் இட் வில் ஜெனரேட் எலக்ட்ரிக்கல் சிக்னல்ஸ். ஸோ என்ன, பிளாஸ்மா மெம்பரேனோட இன்னர் சைடு, பிளாஸ்மா மெம்பரேனோட அவுட்டர் சைடு வந்து எப்படி இருக்கும்? இன்னர் சைடில் ஃபுல்லாக பொட்டாசியம் இருக்கும், அவுட்டர் சைடில் ஃபுல்லாக சோடியம் இருக்கும். ஸோ இதனால என்ன ஆகும்? த பாசிட்டிவ் அண்ட் த நெகட்டிவ் சைடு. ஓகே, பிளாஸ்மா மெம்பரேனுக்கு ஒரு பக்கம் பாசிட்டிவ் சோடியம் பொட்டாசியமும், அடுத்த பக்கம் மோர், தட் இஸ் 3 molecules ஆஃப் சோடியம், 3 மடங்கு சோடியம், 2 மடங்கு பொட்டாசியம். ஸோ இதனால என்ன ஆகும்? ஒரே சார்ஜா இருந்தாலும் there is a difference in the number of charges. So this difference in the number of charges will cause a gradient. என்ன gradient? Electrical gradient. So this difference in the electrical gradient will create a potential gradient or a potential difference. So இதுதான், this only gives the electrical nature of the plasma membrane. Okay, the electrical property of the plasma membrane is maintained like this. So this will only lead to hyperpolarization, depolarization, etc. Okay, so which is very important for the movement of the membrane. Number three, it maintains the tonicity of the ECF. So tonicity, ECF ஓட tonicity or the concentration maintain பண்ணலனா என்ன ஆகும், செல் ஒண்ணு இல்ல shrink ஆயிடும், இல்லனா swell ஆயிடும். ஸோ ஸ்வெல் நார்மல் கண்டிஷன்ல அப்படி நடக்குதா, இல்ல. ஓகே, நார்மல் கண்டிஷன்ல செல்ஸ் நார்மல், எல்லா செல்ஸும் நார்மலா இருக்கு. ஆர்பிசி விச் இஸ் ஃப்ளோட்டிங் இன் த பிளட் இஸ் ஆல்சோ நார்மல். ஆனா அந்த பிளட்டோட டோனிசிட்டி, பிளட்டோட கான்சென்ட்ரேஷனும் எக்ஸ்ட்ரா செல்லுலர், பிளட் இஸ் an extra cellular fluid. Okay, interstitial fluid is also an extracellular fluid. Okay, so blood carries the molecules or the RBC through a blood vessel, whereas the intracellular fluid is bathing all the cells which is present in between the cells, so it makes close contact with the cells. So that in the extracellular fluid, both the interstitial fluid and the blood oda concentration சரியா மெயின்டைன் பண்ணலனா என்ன ஆகும், செல்லோட டோனிசிட்டி பாதிக்கும். ஓகே, செல்ஸோட டோனிசிட்டி பாதிக்கும், இசிஎஃப் ஓட டோனிசிட்டியும் பாதிக்கும். இந்த பம்ப் தான் இதை மெயின்டைன் பண்ணுது. ஓகே, அப்போ என்ன ஆகும், இல்லைன்னா ஐசிஎஃப் இசிஎஃப் ஓட நார்மல் கான்சென்ட்ரேஷன் வில் பி அஃபெக்டட். Okay, then it will lead to some problems, kidney problems, problem in homeostasis, etc. Okay, or else the cells will become shrunk or it will get swollen. So அதுதான் என்ன நடக்குது, இன்ஃப்ளமேஷன்ல அப்படிதான் நடக்குது. So because of some pathogenic invasion, that will happen. Next, to maintain, as I told you, normal tonicity on each side of the plasma membrane. Plasma membrane இந்த பக்கம் செல் இருக்கு, அந்த பக்கம் interstitial fluid இருக்கு. அது மெயின்டைன் பண்றது இந்த sodium potassium pump தான். Next, sodium potassium pump ensures that cell, நான் சொன்னேன் இல்லையா, cells neither shrink nor swell due to the movement of water by osmosis. செல் shrink ஆகிறதோ swell ஆகிறதோ எப்படி நடக்குது, movement of water தான். வாட்டர் உள்ள வந்துட்டா ஸ்வெல் ஆயிடும், வெளியில போயிட்டா, அதிகமா வெளியில போயிட்டா ஷ்ரிங்க் ஆயிடும். ஸோ இதெல்லாம் பண்ணாம இருக்கிறது வந்து இந்த சோடியம் பொட்டாசியம் பம்ப்னால தான். இந்த சோடியம் பொட்டாசியம் பம்ப் வில் மெயின்டைன் த நார்மல் லெவல் ஆஃப் சோடியம் அண்ட் பொட்டாசியம் in the ECF and the ICF. Okay, next what we are going to see is the secondary active transport. நம்ம sodium potassium pump தான் is the best example for primary active transport. So in the secondary active transport என்ன நடக்குது, energy stored in the sodium or hydrogen concentration gradient. So இந்த நம்ம பார்த்தோம் இல்லையா, இந்த concentration gradient, இந்த plasma membrane இந்த பக்கம் அந்த பக்கம் இந்த concentration gradient இருக்கிறதுனால there is some energy which is stored in the ions. சோடியமா இருக்கட்டும், ஹைட்ரஜனா இருக்கட்டும், there is some energy stored in the ions. This energy is used to drive other substances, not the sodium and the potassium, other substances across the plasma membrane against the concentration gradient. Okay, so ATP ல இருக்கிற எனர்ஜி யூஸ் ஆகுது, அதே மாதிரி சோடியம் or hydrogen gradient across the plasma membrane ல இருக்கிற எனர்ஜி யூஸ் ஆகுது. So because the sodium or hydrogen gradient is established by primary active transport. So இந்த gradient எப்படி மெயின்டைன் ஆகுது, எப்படி எஸ்டாப்ளிஷ் ஆயிருக்குன்னா because of the primary active transport. Okay, because of the sodium potassium pumpனால symport and the antiport-னால இந்த மாதிரி மெயின்டைன் ஆகுது. Okay, so secondary active transport indirectly uses energy obtained from the hydrolysis of ATP. நான் சொன்ன மாதிரி not directly, it indirectly uses the energy from the ATP. So some of the stored energy of the sodium potassium pump can be converted to kinetic energy. So என்ன, இந்த sodium potassium pumpல இருக்கிற stored energy வந்து kinetic energy-ஆ convert ஆகுது. Kinetic energy-னா என்னது, energy in motion. Okay, static energy, kinetic energy. So when the molecule is in motion, it is called as the kinetic energy. So in the kinetic energy is used to transport other substances against the concentration gradient. So this is what we are going to see in secondary active transport. Okay, so here also a carrier protein, okay, simultaneously binds to sodium and another substances that change its shape, so that both substances cross the membrane at the same time. So இங்கேயும் sodium potassium pump மாதிரியே sodium potassium pump symport இல்லையா, ஒரு டைரக்ஷன் antiport, சோடியம் ஒரு பக்கம் பொட்டாசியம் ஒரு பக்கம். அதே மாதிரி இங்கேயும் some carrier protein is involved which binds to sodium ஒரு பக்கம், not instead of potassium something else. Okay, some other substance on the other side. So the binding will cause a change in the shape of the pump or the protein, so which will help in the transport of the substance at the same time. Okay, at the same time. So So இது வந்து it is of two types. They are called as the sympoters and antiporters. So if these two transporters அதாவது அதுக்கு சோடியம் பொட்டாசியம் பம்புக்கும் இதுக்கும் என்ன டிஃபரன்ஸ்னா, சோடியமும் பொட்டாசியமும் ஒரே டைம்ல டிரான்ஸ்போர்ட் ஆகல. ஃபர்ஸ்ட் சோடியம் வெளியில போயிடும், வெளியில போனதுக்கு அப்புறம் தான் பொட்டாசியம் பைண்ட் பண்ணி உள்ள வரும். ஆனா இங்க வந்து the transport of the molecules take place at the same time. Okay, that is the difference. So here when two substances move in the same direction, it is called as sympoters. Sim means same side, same. Okay, so transport or movement of two substances in the same direction are called as sympoters. Whereas when the two substances move in opposite direction across the plasma membrane, it is called as antipoters. Antina against simna same. Okay, so there are two kinds of transport: sympoters same side, antipoters opposite side. Now, plasma membrane contains several antipoters and sympoters that are powered by the sodium. Sodium gradient தான் இந்த sympoters-க்கும் antipoters-க்கும் energy கொடுக்குது. Okay, for example என்ன பார்த்தா calcium. Okay, concentration of calcium ion. So உள்ள வந்து சைட்டோசால்குள்ள calcium வந்து concentration கம்மியா இருக்கும். ஆனா for various metabolic reactions, for example muscle contraction எடுத்தா கூட or for example signal transduction இந்த மாதிரியான ரியாக்ஷனுக்கு கால்சியம் தேவை. ஓகே, ஸோ அந்த தேவைப்படும்போது the calcium will be brought inside. Okay, brought inside how? By the antipoter called as the sodium calcium antipoter. So sodium calcium antipoter என்ன படும், சோடியமை வந்து வெளியில அனுப்பும், கால்சியமை உள்ள அனுப்பும். It will transport sodium outside against the concentration gradient and calcium inside whenever it is necessary. So calcium pump. Next வந்து sodium hydrogen antipoter. So first sodium calcium antipoters பார்த்துட்டோம். இப்போ வந்து sodium hydrogen antipoters. This will help to regulate the cytosol pH. So pH எப்படி regulate ஆகுது? By the presence of H plus ions. H plus ions அதிகமா இருந்தா acidic pH. OH minus ions அதிகமா இருந்தா alkaline pH. Okay, so this will help by expelling the excess hydrogen. So this antipoter helps regulate the cytosol's pH by expelling excess hydrogen will be exported out. By contrary, dietary glucose and amino acids are absorbed into cells that line the small intestine by sodium glucose and sodium amino acid sympoters. So நான் சொன்ன மாதிரி digestive tractல small intestine till the stomach, the small intestine performs the enzymatic function, digestion of the food products, that is conversion of the complex organic food materials like carbohydrate, protein, lipid into individual components like glucose, amino acids, fatty acids and glycerol. Okay, so after it has been digested, that is what is called as the digestion. Okay, it has to be absorbed. So the absorption is done from the small intestine. So for the absorption of glucose and amino acid, there are some pumps present in the digestive tract in the small intestine. So those pumps are called as sodium potassium pump, sodium amino acid pump. Okay, they are acting as sympoters. So sympoters அப்படின்னா sodium and glucose will be carried from the digestive tract into the blood. Okay, that is will be absorbed through the wall of the small intestine. So on the wall of the small intestine contains some pump called as sympoters, which will transport sodium and glucose from the small intestine to the blood. Okay, similarly sodium amino acid sympoters, all are sympoters. These two are sympoters because the sodium as well as the amino acid are transported from the small intestine to the other side. Similarly sodium and glucose are transported, that is how they are being absorbed. Okay, so here sodium ions are moving down their concentration gradient while the other solute move uphill against their concentration gradient. So keep in mind that all these sympoters and antipoters can do their job because sodium potassium pump maintains a low concentration of sodium in the cytosol. So இங்க என்ன நடக்குது? The sodium ions are moving down their concentration. Okay, high concentration and then low concentration க்கு move ஆகுது. Whereas the other solutes are moving uphill against their concentration gradient. Okay, So the energy is supplied by the sodium potassium pump. So because of this energy, that is ATPase, the hydrolysis of the ATPase, these sympoters and antipoters can work. Okay. So this is how the antipoter and sympoter. Example for antipoter, rend example pathom. What is the example? Sodium calcium pump and sodium hydrogen pump. See here you can see. So this is the cytosol and this is the extracellular fluid. See, sodium is transported inside and calcium is transported outside. Okay, here sodium is transported inside and hydrogen is transported outside. So here what happens? The hydrogen concentration, if it is more in the cytosol, it will result in that is acidic state. So in order to remove the excess of hydrogen, this particular pump works here. Okay, where the sodium is transported inside. The sodium potassium pump ல என்ன பார்த்தா sodium is expelled outside. ஆனா இங்க வந்து sodium is transported inside and hydrogen is transported outside. But in certain calcium pump, what will happen here? It is shown that calcium is transported outside. Okay, but in some cases calcium is very much required by the cytosol. So in that case, calcium will be transported in and sodium will be transported outside. Okay, so here this is an example for antipoters. And sympoters, you can see this is the extracellular fluid and this is the cytosol. So what happens? Sodium and the glucose are transported towards the same direction. Here sodium and the amino acids are transported towards the same direction. So these are some examples and picture for secondary active transport. Now we have seen primary active transport, we have seen secondary active transport. Okay, that is the inactive transport. In primary transport, we have seen about diffusion and osmosis. So apart from passive transport and active transport, there are so many other kinds of mechanisms by which molecules are transported. So one such mechanism is here transporting vesicles. So vesicles நம்ம வந்து முன்னாடி தெரியும் நம்மளுக்கு. Vesicles are small sac like structures. Okay, vesicles நான் அதுக்குள்ள கொஞ்சம் liquid மாதிரி இருக்கும். Okay, so that liquid will contain some substances which are useful for hydrolysis. For example, lysosome இருக்கும். Okay, hydrolyze enzyme இருக்கும். Okay, so these vesicles, the vesicles are called as little blister or bladder. So these vesicles are present in the cytosol. Okay, so they are called as spherical sac, small round shaped sac which are called as blister or bladder. Okay, so variety of substances are transported in the vesicle from one structure to another within the cell. So in the vesicle மூலமா many different kind of substances, அதாவது by a process called as endocytosis and exocytosis. Okay, இந்த that is cell molecule வந்து non-polar organic hydrophilic hydrophobic ஓட transport எல்லாம் நம்ம பார்த்தோம். So passive transport will carry the hydrophobic non-polar molecules and slightly polar molecules across the plasma membrane, whereas the passive transport will carry or transport the charged polar molecules across the plasma membrane with the help of some integral proteins present in the plasma membrane. But சில molecule வந்து இது ரெண்டு மூலமாவும் transport பண்ண முடியாது. ஸோ அதுக்கு என்ன பண்ண முடியும் அப்படின்னா by a process called as endocytosis. எண்டோசைட்டோசிஸ்னா வெளியில இருக்கிற பொருளை உள்ள கொண்டு வர்றதுக்கு எண்டோசைட்டோசிஸ் will be used. எக்ஸோசைட்டோசிஸ் உள்ள ஏதோ ஒரு வேஸ்ட் இருக்கு, ஓகே, அதை வெளியில தள்ளணும் அப்படின்னா எக்ஸோசைட்டோசிஸ். ஸோ அதுக்கு வந்து இந்த வெசிக்கல் sac like structure is very very useful. So these vesicles are present. Okay, so they will bind or get attached with the plasma membrane. So at times it will perform endocytosis process or exocytosis process. So by which the materials can be either transported inside by endocytosis or outside by exocytosis. Okay, so during endocytosis nala within materials move into the cell in a vesicle formed from the plasma membrane. So main என்ன ஆகும், இந்த vesicle வந்து it is used for transport of these substances. So the vesicles will bind to the plasma membrane and ஒரு invagination நடக்கும். எண்டோசைட்டோசிஸ்ல வந்து அந்த plasma membrane ஒரு invagination உள்ளுக்கா அது வந்து வளைஞ்சு ஒரு இது மாதிரி ஸ்ட்ரக்சர் மாதிரி கொண்டு வந்து it will carry the vesicle inside. So this vesicle will pinch off from the plasma membrane and it will bind to the lysosome. okay, and it will transport. Okay, that is another process called as endolysosome. Okay, so phagocytosis. So phagocytosis is another method where exocytosis is involved. So materials from the cell, okay, will be carried in the vesicle. This vesicle will fuse with the plasma membrane, and then by a process called as exocytosis, அதாவது வெளியில உள்ள இன்வாஜினேஷன் மாதிரி இது வெளியில வந்துட்டு ப்ரொட்ரூட் ஆயிட்டு அமீபா லைக் மூமெண்ட் ப்ரொட்ரூட் ஆயிட்டு it will pinch off from the plasma membrane and it will go into the extracellular fluid. Okay, so they require energy, they also require energy that is supplied by the ATP. Okay, so in the transport by vesicle is also an active process. So unlike the passive, this is also a process by which the molecules can be transported, and it is an active process, it requires energy. Now, what is endocytosis? So, endocytosis is another invagination inside. There are three types of endocytosis. One is receptor mediated endocytosis, number two is phagocytosis, and number three is bulk phase endocytosis. So, that is what we are going to see now. Now, what is receptor mediated endocytosis? This is the receptor mediated endocytosis. So, what we are going to see now. So, it is a highly selective type of endocytosis, okay, by which cells take up specific ligands. ஸோ செல்ஸுக்கு ஏதாவது லிகான்ஸ் தேவைப்பட்டதுன்னா அதை வந்து எப்படி உள்ள கொண்டு வர்றது, ஓகே, அதுக்காக இந்த ரிசெப்டர். ஸோ ரிசெப்டர்ஸ் ஆர் present on the plasma membrane. So the ligands can bind to the receptors. So there are this binding of the ligand and receptor are specific. Okay, so ஒரு லிகாண்டுக்கு specific receptor molecules are present on the plasma membrane. So the ligands will bind to the plasma membrane and will be taken inside. இங்க வந்து a vesicle forms after a receptor protein in the plasma membrane recognizes and binds to a particular particle in the extracellular fluid. So when a ligand binds to the receptor, என்ன ஆகும், உள்ள வந்து cell will recognize, okay, ஒரு ligand bind ஆயிடுச்சு. ஸோ என்ன பண்ணலாம், ஒரு வெசிக்கல் இமீடியட்டாக ஒரு வெசிக்கல் ஃபார்ம் ஆகிடும். ஓகே, ஸோ அந்த வெசிக்கல் ஃபார் எக்ஸாம்பிள் இங்கே என்ன கொடுத்துருக்காங்க அப்படின்னா கொலஸ்ட்ரால். ஸோ செல்ஸ் டேக் அப் கொலஸ்ட்ரால் containing LDL, transferrin, an iron transporting protein in the blood, some vitamins, antibodies, and certain hormones by this particular method, receptor mediated endocytosis. So எப்படி இந்த LDL, transferrin, vitamins எல்லாம் எப்படி உள்ள போகுது, antibodies, hormones எல்லாம் எப்படி உள்ள போகுதுன்னா by receptor mediated endocytosis. So this LDL, this transferrin, vitamin, antibody will act as ligand. So for these ligands there will be specific receptors present on the plasma membrane. So when they bind, the cell will recognize the binding and immediately it will form a vesicle. So here you can see how the thing happens. Okay, so binding of the receptor LDL complex. See, LDL particle receptor. Now it is bound. Okay, so it forms a clathrin coated pit. Okay, ஒரு பிட் மாதிரி ஃபார்ம் ஆகுது. See, ஒரு பிட்னா என்ன சொல்றது, ஒரு பள்ளம். Okay, now this undergoes an invagination. Now the plasma membrane is forms an invagination. உள்ளுக்கா இது வந்து ஒரு குழி மாதிரி பிட் மாதிரி ஃபார்ம் ஆகுது. மேலே நீங்க பாத்தீங்கன்னா you can see the receptor and a golden color ligand, that is the LDL. Now see how this invagination widens and pinches off from the plasma membrane. Plasma membrane seals. Okay, plasma membrane குள்ள ஓட்ட போட்டா எதுவுமே வந்து நடக்காது. Okay, it was because I told you know the lipid phospholipid bilayer வந்து it is a very fluid mosaic model இல்லையா, it is very flexible and it keeps moving from one place to another. ஆனா அதனால தான் this method of in vitro fertilization. In vitro fertilization என்ன பண்றாங்க, ஓவம்ல வந்து sperm inject பண்றாங்க. Okay, that is you call it as micro injection. So micro injection ஓவம் வந்து இன்சர்ட் பண்ணாலும் ஓவம் வந்து ஓட்டை ஆகாது. ஓகே, ஒரு நீடில் வச்சு உள்ள நுழைச்சா கூட நீடில் எடுத்த பிறகு immediately the plasma membrane will seal. Okay, will seal off so that it does not form ஒரு ஓட்டை இருந்தா என்ன ஆகும், ஒரு பலூன்ல ஓட்டை போட்டா உள்ள இருக்கிற காத்தெல்லாம் வெளியில வந்துடும். அதே மாதிரி ஒரு தண்ணி டேங்க் இருக்கிற இடத்துல ஓட்டை போட்டா என்ன ஆகும், தண்ணி எல்லாம் வெளியில வந்துடும். ஆனா this plasma membrane has got a specific property where even when you puncture the plasma membrane, okay, it will immediately seal the hole or the puncture. Okay, so here you can see After the pinching of the vesicle, the plasma membrane on the top will immediately be sealed. Okay, now the third process, you can see this is the vesicle. So vesicle kulla enna irukku, it is the clathrin coated vesicle. So ulla vandu receptor, so velila irukkiradella it is called as clathrin. Okay, you can see these are the clathrin. Okay, so ulla vandu receptor oda LDL bind irukku. Now what happens here, the outer coating is, okay, the clathrin coating is removed. இங்க பாத்தீங்கன்னா this is coated and this is uncoated, so it is removed. So this is the uncoated vesicle. Now the vesicle fuses with the endosome. So there is an endosome here. Okay, so now the vesicle will fuse with the endosome. So what will happen, see, you can see the LDL is transported. Okay, so here the receptor, மீதி இருக்கிற receptor அல்லது the receptor and the ligand will detach. So இங்க வந்து fused-ஆ இருக்கு ligand and the receptor, இங்க receptor தனியா போயிடுது, ligand தனியா வந்துடுது. So அந்த receptor எல்லாம் வந்து திருப்பியும் வந்து recycle ஆகும். Okay, so what will happen, it will be again taken by the endosome. Okay, and it will be carried to the plasma membrane, where they will go and attach themselves on the outside of the plasma membrane to receive more ligands. These ligands will be entrapped in a vesicle, okay, called as this is a transport vesicle. Now these ligands will be carried, okay, carried. See, you can see how it is carried. Here on the it binds with the lysosome and undergoes degradation. Okay, see, this is the lysosome, this is the lysosome, and this endosome vesicle binds with the lysosome to form the endolysosome. Okay, so lysosome la enna irukku, it contains enzyme called as hydrolase. Okay, this hydrolase will degrade the LDL and it will be utilized for the whatever maybe the cholesterol will be utilized. LDL is made up of cholesterol, triglycerides, and so many other things. So they will be degraded and individual components will be utilized. Okay, this is what now we will see what are the different steps involved in the receptor mediated endocytosis of the LDL. Okay, first binding. So on the extracellular side of the plasma membrane, an LDL particle that contains cholesterol binds to a specific receptor in the plasma membrane to form a receptor LDL complex. So first LDL வந்து blood ல இருந்து செல்லுக்கு transport பண்ணணும். So here LDL is the ligand. So this ligand binds to a specific receptor. So and the receptor எங்க இருக்கு, plasma membrane மேல இருக்கு. So when it binds, it forms a receptor LDL complex. So the receptors are integral membrane proteins. So you know what are integral membrane proteins that are coated in regions of the plasma membrane called clathrin coated pits. So எங்க இருக்கும், இந்த clathrin coated pits கிட்ட தான் இந்த வந்து receptors இருக்கும். Okay, so the protein, a protein called clathrin attaches to the membrane. நம்ம பார்த்தோம் இல்லையா, இதெல்லாம் clathrin. So this clathrin protein, this is a clathrin coated vesicle. Okay, so this attaches, so the clathrin, a protein called clathrin attaches to the membrane on its cytoplasmic side. So அந்த பக்கம் receptor, இந்த பக்கம் cytoplasmic side வந்து clathrin. So many clathrin molecules come together forming a basket like structure around the receptor LDL complex. This will cause the membrane to fold inside. Next வந்து formation of vesicle. Now in the invaginated membrane வந்து என்ன ஆகும், will be pinched off from the plasma membrane. Okay, around the clathrin coated pit. இந்த clathrin coated pit என்னது, cytoplasmic sideல வந்து clathrin இருக்கும், வெளியில வந்து you have the receptor. Now this will be pinched off and small piece of the membrane pinches off. This resulting vesicle is known as the clathrin coated vesicle. Now the clathrin coated vesicle is free to float into the cytoplasm. இதுக்குள்ள என்ன இருக்கு, LDL வந்து ரிசெப்டரோட காம்ப்ளெக்ஸ் ஆயிட்டு ட்ராப்டா இருக்கு. நெக்ஸ்ட் வந்து என்ன நடக்கும், இந்த clathrin வந்து ரிமூவ் ஆயிடும், uncoating. So immediately after the vesicle is formed, the clathrin coated vesicle loses its clathrin coat to become an uncoated vesicle. அப்ப இந்த clathrin எல்லாம் என்ன ஆகும், திருப்பியும் it will get transported. See, it will be transported to the plasma membrane where it reforms the see இங்க clathrin coated, உள்ள clathrin மேல வந்து receptor, இந்த மாதிரி திருப்பி போய் ஜாயின் ஆயிடும், plasma membrane ஓட ஜாயின் ஆயிடும். Now fusion with endosome. இந்த uncoated clathrin remove ஆனதுக்கு அப்புறம் இந்த uncoated vesicle என்ன பண்ணும், immediately fuses with the another vesicle called as the endosome. Okay, so endosome oda fuse ஆயிடும். Fuse ஆயிட்டு என்ன ஆகும், the LDL particle வந்து receptor கிட்ட இருந்து separate ஆயிடும். So LDL which is bound to the receptor complex will get separated. Okay, in the receptor வந்து again will be reused. It will go and bind to the plasma membrane. That is what is called as the recycling of receptors. So most of the receptors accumulate in the elongated protrusion of the endosome. They pinch off, forming transport vesicle that return the receptor to the plasma membrane. So this LDL வந்து is returned to the plasma membrane about 10 minutes after it enters the cell. Now degradation in lysosome. So இன்னொரு vesicle இருக்கு, other transport vesicle which contains the LDL particle. That is bud off the endosome and soon fuse with the lysosome. This endosome will fuse with the lysosome which contains the enzyme hydrolase. Hydrolase is a digestive enzyme. This hydrolase will break the LDL. It will be break the LDL particle into amino acid, fatty acid, and cholesterol. Okay, so these molecules then leave the lysosome. The cell uses makes use of the cholesterol எதுக்காக for synthesis of steroids and membrane cholesterol. Okay, and some other steroidogenic molecules. Fatty acid and amino acids present in the LDL will be used for, fatty acids will be used for ATP production, amino acids will be used for, protein synthesis. So this is what is happening in receptor mediated endocytosis of a low density lipoprotein particle. Did you understand any doubt in this? Are you there? Students?
Yes, ma'am.
Time ஆயிடுச்சா? 40 ஆயிடுச்சுல? Ma'am is waiting for another class. Pardon? Waiting. Oh, I'm sorry, I didn't recognize the time. Okay, next two thing is phagocytosis and bulk phase endocytosis. Okay, tell ma'am my sorry. We will end up the class now. We will continue with the phagocytosis and bulk phase endocytosis in the next class. Okay?
Okay, ma'am.
Okay. Okay, ma. Thank you.
Thank you, ma'am.
Okay, ma.
Thank you.
Okay.